So vaccines normally require years of testing and much more time to produce tons of them. But as we know, scientists and the world are hoping to develop a COVID-19 vaccine ASAP. But how are these vaccines tested? The exploratory stage involves basic lab research and often lasts between 2 to 4 years. I can get into the whole sciencey stuff, but we're not going to understand anyway. At this point, researchers give the vaccine to animals to test its safety and see if it triggers an immune response. This gives researchers an idea of the cellular responses they might expect in humans. So this stage has 3 phases. Fase número 1. The vaccine is given to a small group of people to determine whether it is safe and to learn more about the immune response it provokes. The vaccine is given to hundreds of people so scientists can learn more about its safety and correct dosage. The vaccine is given to thousands of people to confirm its safety, including rare side effects and effectiveness. These trials involve a control group which is given a placebo. After a successful phase 3 trial, the vaccine developer will submit an application to the FDA. Then the FDA inspects the factory where they'll make it and approve its labeling. And the last 2 stages are manufacturing and quality control. So what's happening right now with the COVID-19 vaccine? Here's a little update from the amazing Dr. A.
We have over 150 horses in this race, over 150, but four are leading the pack. We have CanSino Biologics based out of China. We have Moderna based out of Cambridge, Massachusetts. We have AstraZeneca and the Oxford Group, and then we have Pfizer. All 4 of these candidates have passed phase 1, they've passed phase 2, they've shown you get an antibody response, they've shown safety, they've shown an antibody response, and then phase 3, they are now all in the beginning stages of starting their phase 3 trials over these weeks to come. So that is exciting news. They're in the final and 3rd phase of vaccine development, all hoping for potential results of effectiveness, that it actually works by the end of the year. But there are 3 major lingering questions very important to talk about here and break down. 1, recent studies, several, have now shown that antibodies quickly decline once a person's infected. By 50 to 70%, the antibody levels potentially decline within just months after an infection. So a huge question here is, if a natural infection does not produce long-lasting antibodies, will bits and pieces of a dead infection in a vaccine do it? A huge question that we don't know. 2, because of this reality, will we need multiple doses or vials of the vaccine? Will it need to be a 1 or 2 or 3 booster shot? Look at hepatitis B. It's a 3 booster shot in total in order to generate enough antibodies. If that's the case with the COVID vaccine, and let's say we need 3 shots for it, that's not only 300 million vials for 300 million Americans, not 600 million vials. That is almost 1 billion vials, 900 million vials for a 3 booster shot, if needed, to generate robust enough antibodies. And so that's a huge question. Will we be ready to produce 900 million, if needed, vials of the vaccine in time to actually immunize the United States population? And then 3, if all else fails, if vaccines do not work, look at HIV. It's been around for more than 4 decades, 40 plus years, and there is still no vaccine to HIV. 40 years. So if there is no vaccine to COVID, just like there are no vaccines here for the normal coronavirus colds that we also get, then do we need to start really focusing on treatments, preventative treatments that we can take, or treatments we can give at the very beginning of symptoms that prevent any hospitalizations or ICU stays or breathing tubes, functionally converting COVID-19 into the common cold? This is what UCSF researchers are now focusing on because they don't see as much hope around the vaccine pathway as potentially the treatment pathway.